So guidelines for estimating space use. Okay, so similar to the set of slides for behavioral state estimation, I'm going to define a set of guiding questions that you may want to consider when trying to choose one method over another. So to start off, what's your sample size? So sample size is going to be really important, and some methods are going to be more sensitive to sample size than others. This may impact how you interpret your results, whether they're reflective of the actual distribution of all individuals of a population, or maybe the number of observations for a particular individual, and whether that's reflective of its home range versus just the brief area used during the tracking period. Additionally, another thing I've mentioned a lot during these different topics today is what's the sampling frequency. So that'll greatly impact potentially which method you should choose, and the relationship between your points and any autocorrelation that's present. Are you trying to detect temporal patterns? So just estimating overall space use for an entire tracking duration is something that most people do a lot of times. But there are plenty of studies where they're interested in these temporal patterns or shifts in space use, whether that's diel patterns, seasonal patterns, annual patterns. That will also kind of impact what method you may want to use. What's the general movement pattern exhibited by your individuals? So like we just saw at the end here with this comparison, for the resident individuals, it may not really matter which method you use, because given the sample size and their movement pattern, all the methods performed relatively comparably. But for the migratory individuals, there was a large discrepancy in the areas used and estimated being used by these different methods. So that should also be taken into consideration. Are you trying to estimate the occurrence or the range distribution? So I touched on this earlier today, but essentially, are you trying to estimate the area that the animal already used when you tracked it? Or are you trying to also allow for a potential future use that it hasn't used yet, that you want to account for its total, let's say, home range? Is that of interest to you? And lastly, something I didn't cover during any of the different components today, but I think is a really important area in this topic moving forward, is the incorporation of the effects of environmental covariates. So obviously an animal's not moving in space independent of any behavioral considerations or habitat considerations. Those are definitely drivers of how they use space. Everything we've considered today assumes that those are basically negligible or doesn't account for them at all. But there are a number of different methods that do account for the effect of environmental covariates in different ways on the space use of these different organisms. And again, this comparison to the right between or among a lot of these different methods that we showed today along the rows, and then the columns of the different time intervals. So depending on the time interval and then the movement pattern, certain methods may perform better than others. So first, what is your sample size? So modern telemetry devices generally collect high levels or amounts of data, and it's generally assumed, at least by me, that researchers will collect probably over 100 observations per individual, and often more than one observation per day, unless there's like a tag malfunction or something else goes wrong. Unlike traditionally, when a lot of these methods were originally developed with radio telemetry, and then you had much smaller sample sizes, where the original proposal was you needed at least 50 observations with radio telemetry to get valid estimates from kernel density estimation. And that's not a problem anymore. Now we have to deal with autocorrelation due to high sampling frequencies from our tags. So again, I want to emphasize here at the beginning that these aren't hard and fast rules. These are just general rules of thumb that you may want to think about, that are relatively subjective in terms of what sample size is going to impact what method you use. But let's say all of your observations across all individuals totals over 1,000 total like points or observations. Then the methods you may want to consider If you're going to use kernel density estimation, to use the plugin bandwidth estimator, or to use dynamic Brownian bridge movement models. Typically, once you start getting to very large sample sizes, the least squares cross validation method for the bandwidth estimator of kernel density estimation starts not performing well and may be hard or difficult to estimate that bandwidth. So in general, maybe better to use the plugin estimator by comparison. If the sample size is maybe roughly smaller than 1,000, again, this number is just subjective. If it's on the smaller end, then you can probably use either the plugin or the least squares cross validation method, or the dynamic Brownian bridge movement model. And notice here I'm omitting minimum convex polygons. And this is going to depend on your project goals. So you may need to consider whether the estimated UDs are performed for the population at large, like what we initially did for the kernel density estimates, or if you want them to be estimated per individual separately. That'll impact maybe what sample size you need to consider to produce these UDs. The one thing that's been brought up more frequently, or brought up as a point of contention more recently, is that how do you know if you have enough animals tracked to characterize the population utilization distribution well? So that might be hard to kind of define, especially when people are trying to come up with guidelines for designing a study, how many tags they need to put out, because you may not know how long the tags are going to stay on, how many points you're going to get per individual, where those individuals are going to move. So there's all these considerations. But this recent paper by Shimada and colleagues from last year has this proposed method, which I think is very similar to a rarefaction curve in community ecology, looking at species diversity, whether it's the diversity of an ecological community, or you're looking at, let's say, a dietary index, and you're looking at like stomach contents per se, and you're trying to see if you've sampled enough stomachs to characterize that diet properly. Essentially, you want to use a method where you're randomly including different animals of your tracked population to see how much area they've used, and then measure the overlap among those separate individual utilization distributions. And once you get to a point where, once you keep adding more individuals, if they overlap like with 100% probability, you've probably defined the population's utilization distribution pretty well. So this is showing two examples here on the left where we have these curves, and we essentially wanted to reach an asymptote, showing that we've sampled enough individuals to estimate the population utilization distribution well. If the area keeps increasing as you add more and more individuals, you probably need to continue sampling more individuals. And this is kind of shown in a spatial context here on the right, where each of these separate subplots shows a different subset of the data. So we have n equals 4, a sample size of 4 individuals in this plot on the left, showing one region that's being used by these different turtles, in this case, flatback turtles off of Australia. In the middle plot, sample size has been increased to 11, and we've seen this expansion northward of this total population utilization distribution. And if you increase this to, let's say, 29 individuals, it actually hasn't really changed that much at all. Maybe the intensity is increased in a little bit in some areas, but it's essentially been unchanged. So probably around 11 individuals, or maybe a little bit more, is the minimum number you would need to properly estimate the distribution of this subpopulation in this area or this part of the world. Second, so what's the sampling frequency? What's the time interval? So higher sampling frequencies are obviously associated with higher autocorrelation in your data. So if the time interval is less than 24 hours, which again, I would argue is essentially all modern telemetry data sets, whether they're satellite or acoustic, you need to somehow account for some form of temporal and spatial autocorrelation, which is somewhat implicitly done by the dynamic Brownian bridge movement model, when it's using this behavioral change point analysis to estimate these shifts in the Brownian motion variance. So in general, I'm recommending if you're using some form of modern telemetry right now that's not radio telemetry, that you move away from kernel density estimation and use a method such as dynamic Brownian bridge movement models. If you are going to use some form of kernel density estimation, then I highly recommend trying to use the autocorrelated kernel density estimation method, which directly accounts for or explicitly accounts for this temporal autocorrelation. However, again, I need to mention that this method is estimating the range distribution, so it's not estimating the occurrence distribution. But if your time interval is longer than 1 week, so like if you are still doing radio telemetry, you probably can do kernel density estimation because these points would be probably considered independent of one another. And again, this is highlighted from this plot or this figure I showed previously, where we have these tracks that are shown at a 1 hour time interval for a buffalo and a 1 week interval, and the comparison between AKDE versus KDE. Okay. Third, are you trying to detect temporal patterns? So this might depend on whether you're estimating the UD at the individual or population level. So if you're looking to compare, make these comparisons within a single individual, ideally would be tracked across this temporal scale of interest. So if you're looking at diel patterns, it needs to be tracked over both night and day. If it's tracked over, or you're interested in seasonal patterns, you want it to be tracked over multiple seasons, ideally all seasons. If you're looking at a population level, you can probably get away with certain individuals only being tracked for a single season and then comparing multiple individuals. So diel patterns, because of the fine temporal scale, ideally you would probably use the dynamic Brownian bridge movement model. If you're using monthly patterns, you may also be able to use the kernel density estimates in addition to dynamic Brownian bridge movement models. And again, I would in general recommend to never use minimum convex polygons to estimate space use. So an example of seasonal patterns of space use based on kernel density estimates is shown on the right here for hawksbills in the Arabian Gulf. And it may be obvious to tell that these kernel density estimates are actually being produced by a reference bandwidth estimator, which is why they appear quite large compared to the points of these tracks that you can see. So ideally, that would instead be done with either the plug-in method for kernel density estimation or a method such as AKDE or dynamic Brownian bridge movement models. And then, yeah, seasonal patterns, either KDE or dynamic Brownian bridge movement models. And these are highly dependent on the sampling frequency of your tags. And in general, so what's the general movement pattern exhibited by your animal? So this is, again, another consideration that we observed when comparing the migratory turtles versus the resident turtles off Brazil. So resident behavior may be defined by a single cluster of points for a given individual. And this may be a case where kernel density estimation does perform pretty well. And I'm sure that dynamic Brownian bridge movement models would perform fine. If your tracks are performing more ranging behaviors, whether they're producing forays, they're growing out somewhere and then coming back after a brief amount of time, or they're dispersing from one location to another and staying there, or they're performing these long-term migrations where they might go to a breeding ground and then come back to a foraging ground and stay there for maybe years at a time. You probably want to use dynamic Brownian bridge movement models. The KDE methods will probably overestimate the space that's actually being used in these cases. And if they're performing a mix of these different movement patterns, so there's clusters of points, or there's maybe some forays or dispersals or a spread of points, you can probably use either of these methods, KDE or DBBMM. Those should both work in these instances. Additionally, are you trying to estimate the occurrence or range distribution? So it appears to me, based on looking through many studies, that most people are interested in the occurrence distribution. So what space did the animal use during this tracking period? However, if you're trying to plan for conservation, you may be actually more interested in the range distribution. So what areas may the animal use in the future, and using that to inform the proposal of new protected areas. So in general, for occurrence distributions, the kernel density estimates and DBBMMs will actually give you these occurrence distributions. But if you want the range distribution, you may want to use the AKDE method or other types of mechanistic movement models to do so. And then lastly, are you interested in the effects of environmental covariates? So simply, if not, use one of the methods we covered today, KDE or DBBMM. Again, we're not going to be using minimum convex polygons moving forward, and ideally moving away from kernel density estimates if possible. But if you are interested in the effects of environmental covariates, there's a number of different movement methods that are available. And they do so in different ways. There are things such as geostatistical mixed effects models that can account for spatial or spatial temporal autocorrelation as a random effect. And then you can use environmental variables as covariates that impacts the space that's being used, the probability of using a given area. Alternatively, there's other like resource or habitat selection models that estimate habitat preferences, but then use these preferences to derive utilization distributions. So this is another interesting way that I think will be really powerful moving forward for estimating space use that might represent actually the range distribution, since you didn't necessarily see the animal move to all these different locations within the study area. So for the geostatistical mixed effects model, you might have to fit a triangulation mesh like this, and you can even take into account the boundary effects of islands or land if you have a marine animal. And then you can get a surface like something here. So this is showing the predicted probability of occurrence for loggerhead turtles along the Atlantic coast of the US here. And we see a overall prediction of space use. And then we have these separate monthly predictions that may or may not include environmental covariates, depending on your interests. So other methods I did not cover today, but I wanted to mention that you should potentially look into depending on what you're interested in and what your data set looks like. I've mentioned AKDE. We didn't cover it today, particularly because it doesn't perform well necessarily on migrating individuals. But this is something you should definitely consider depending on your study questions and your data sets. Another method I didn't mention at all was local convex hulls. And this is like producing a minimum convex polygon for different subsets of your points. So this is something else that's out there that people are using. There's also movement-based KDE, which I think resembles the dynamic Brownian bridge movement models, also called biased random bridges. And I think it'll probably perform comparably to the DBBMMs. So trying to account for this movement process when estimating a kernel density estimates. And then we get into these other models that can account for environmental covariates or not. So like these geostatistical mixed effects models, continuous time Markov chain models that estimates habitat preferences and then use those to estimate utilization distributions, as well as step selection functions, which do a similar thing. There's also a Brownian bridge covariates model that's been recently developed by the same author that developed the DBBMM that essentially is using environmental covariates to inform this Brownian bridge movement process. And if you have any interest in looking into some of these other methods, you can also look at the useful resources page on the workshop website, and that has a bunch of other links for different tutorials or papers that are related to these things. So with that, that is it for the workshop.